Hello students, in this video lecture we are going to learn about the DNA damage and repairs, right? So in this chapter we will make you understand the importance of maintaining the integrity of DNA, right? So different types of damages they induce in the DNA and their consequences. So if not rectified from the DNA molecules, we will going to describe what will going to happen, right? So the survival of every cell depends the integrity of the genetic material, right? The accuracy which will be maintained by the cells, so that we are going to discuss here. So first we'll see what is meant by DNA damage. So DNA, it can be easily damaged under the normal physiological conditions, right? So we'll see the different physiological conditions which are responsible for the DNA damage. Then the return of the damaged DNA to its normal sequences and structure, it is called as a repair. So in the living cells, we have the repair mechanism to um, convert the damaged DNA into the normal DNA sequences, right? So that is called the repair mechanisms. Okay, so many different kinds of physical as well as the chemical agents that damage the DNA, right? Some of these are like the endogenous agents as well as exogenous agents. So these agents that damage the DNA can be mutagenic or cytotoxic, or they may be mutagenic as well as cytotoxic also. So the DNA damaging agents that cause the mutations, that are called as the mutagens, right? So these mutagens, they causes the changes in the DNA sequences. We will see the different types of DNA damages. So damage done to the DNA by the physical, chemical, environmental agents, they can be broadly classified into the four categories with the different types, right? Okay, so the prokaryotic cells as well as the eukaryotic cells, they have the nucleotide sequences, right? So these DNA molecules, they consist of these nucleotide sequences, and their integrity is important for the survival of any kind of cell, right? So the DNA as well as the RNAs, they can be quickly replaced on damage by using the information which is encoded in the DNA, right? So but the DNA molecules, they are irreplaceable, and hence the damage in them need to be repaired. So in our body, the cells have some kind of DNA repair mechanism. So that DNA repair mechanism will activate to correct such kind of DNA damages occur due to some environmental factors or some chemical mutagens or physical mutagens, right? So the different types of the DNA damages, they include the single base alteration due to the deamination, right? Or depurination, as well as the base alkylation, insertion or the deletion of the nucleotides, as well as the incorporation of base analogs. Then the two base alteration, like the UV light, they induces the pyrimidine dimer alteration. So this causes the thymine dimers, means thymine will pair with the thymine. So its abnormality, it caused due to the exposing of the cells under the ultraviolet light. Then the chain breaks. So what happens, sometimes the oxidative free radicals formation, so they causes the ionizing radiations, and due to that the chain may be break. Then cross linkage, it it takes place between the bases in the same or the opposite strands of DNA, right? Which between the DNA and the protein molecules. So these are the different kinds of DNA damages. First we'll see the single base alteration. So you can see here the cytosine pair with the uracil, but due to the then here you can see the thymine, it's generally paired with the adenine, but due to the deaminations, the 5 methyl cytosine, it may altered, right? And it get bound with the thymine. Then you can see here adenine, it bonded with the, it can convert it into the hypoxanthine, right? So because of this single base alteration, the DNA can be damaged. Okay, here you can see also the spontaneous deamination or depurination or induced uh, mutation which caused by the exposure to the UV light. And you can see here the thymine dimer also, the formation of thymine dimer. Sometimes what happen, the guanine it's undergoes the alkylation, and due to that the sigma methyl guanine get formed. So this is the abnormality in the DNA. Then the double base alteration, right? So due to the exposure in the UV light, so you can see here double base alteration is there. Okay, thymine will bound with the thymine also. So such kind of mutation, such nitrogenous bases is bounded with each other, that is called as a double base alteration, means thymine will pair with the thymine, that is called the cyclobutane thymine dimers. Okay, or 6 for photo products. So DNA damaging agents like the spontaneous agents, they are highly reactive oxygen radicals, right, which produce as a byproduct during the normal cellular respiration as well as by other biochemical pathways. Radioactive species, right? So these ROS, that is radioactive species, radio oxygen species. So these are like the hydrogen peroxide, and then hydroxyl radicals. So these are the most potent. Then the superoxide. So these are the reactive oxygen species, means you can see that this oxygen it's highly unstable, right, and it may interact with the DNA and cause the DNA damage. The reactive oxygen species cause the DNA damage, such as the oxidation of nitrogen bases or deoxyribose or and the strand breaks, right. So you can see the radiation, it can cause the mutation. The most potent, they are the cosmic gamma rays, ionizing radiations. Okay, so the lights, it's get divided into different uh, regions like the ultraviolet, visible or infrared, right. So ultraviolet light, that is UV light, it ranges up to the 400 nanometer, and this light is very dangerous, right. But what happens sometimes, the ozone layers, right. So ozone layers always it filter this ultraviolet light, but in some geographical region at the polar region, as well as due to the due to the continuous pollution, what happen, this ozone layer it get depleted, and thus whatever the ultraviolet light which will be exposed from the, which will be radiated through the sunlight, okay, so that haven't be get filtered by the ozone layers, right, and they can cause the DNA damage, right. So these radiations are high energy electromagnetic radiations, due to the exposure of which the cell experience the considerable damage to their DNA. So these are the ultraviolet light. The major types of damage caused by the ultraviolet light is divided into three types, that is UVA, UVB and UVC. So UVA range is varies from 321 to 400 nanometer. UVB varies from 296 to 320 nanometer, and UVC it ranges from 100 to 295 nanometers, right. So these are the ultraviolet light. They are divided, okay, into the three major kind: ultraviolet A, B and C. So this UV light on the DNA structure, they show such kind of thymine dimers. So here you can see the yellow strips. So they have the thymine have to bond with the adenine, right, but due to the exposure of DNA into the ultraviolet light, okay, so this thymine dimer get formed. Thymine will pair with the thymine, right. So this can be caused due to these ultraviolet rays. Then the chemical agents like the deaminating agents, sodium nitrate, okay, then sodium nitrite, nitrosamine, nitrous acids, right? So these are the deaminating agent, means they causes the deamination, amino group get removed. Or some chemicals like the dimethyl sulfate, that is commonly called DMS, or dimethyl nitrosamine, or nitrogen mustard. So this can cause the alkylation, right? So these are the alkylation agents which can also cause the DNA damage. So due to such kind of DNA damage, mutation can be occur, right? So this mutation, it's sometimes beneficial, sometimes it's dangerous for the living organism. Most of the time it's dangerous, but sometimes it may be helpful also, because we have one single common ancestor, right? Means all the living organisms, they have evolved from the single common ancestor, single living organism. So that single living organism face the mutation, and due to that diversity of the living organism get formed. So this is called as a species evolution of a species, right? So this evolution of species, they may also cause because of the mutation, right? So mutation refers to a change in the DNA structure of a gene, and the substances like the chemical substances which can induce the mutations are collectively known as a mutagens, right? So the changes that occurs in the DNA on the mutation are reflected in the replication, transcription and translation, right? So mutation, it occurs in the two ways: spontaneous mutation and induced mutations. So spontaneous mutation, it can be mistakes happens during the DNA replication, and this is the normal mutation, spontaneous mutation. That's why it is curable. Means if this mistake will be happen, this spontaneous mutation will be happen, then there will be a machinery in our body that is called the DNA repair machinery. So this will going to correct such kind of mutation. So that's why spontaneous mutation is very less harmful for the living organism, but induced mutation, it caused by the mutagens, and it is recently added in the environment because of the different mutagenic chemicals, as well as mutagenic physical agents or environmental factors, right? So this cannot be curated, that is induced mutation. So there are different kinds of mutation, that is point mutation. A point mutation or a single base substitution is a type of mutation that causes the replacement of single base nucleotides with the another nucleotides of DNA. Okay, so the substitutions, they are like the transition or transversion. So first we'll see the transition. In this case, the purines or pyrimidines is replaced by one another, okay, or the transversion. So these are the characterized by the replacement of purines by pyrimidines, or pyrimidines by purines, vice versa. Okay, so transition means what? The substitution will be takes place within the purines, okay, and or pyrimidines. But transversion, it will be it can characterize by replacement of purines by the pyrimides, right? So this substitution, you can see here, the single nucleotide change happens in the DNA. So this DNA which is having the genetic code, okay, in the sequences, that will convert into the mRNA, and that mRNA codon will convert into the single amino acid by the triplet. So that is a CGU. So because of the single change in the substitution here, okay, so what here have to, that is the thymine, okay, so thymine will convert into adenine, and here what will happen, the histidine will be get formed in case of arginine, right? So because of the single mutation, single nucleotide change also, the amino acid can be changed. So these are the different kinds of the genetic codes you already studied in your T.Y.B.Sc classes, right? So these are the codons which consist of three nucleotides, right? That's why they are called as the triplets. So these triplet codons, they are specific for specific amino acids only. So there is a start codon as well as there is a stop codon. Okay, they can be start the translation as well as they can stop the translation process also. Okay, so because of the point mutation here, so sometimes point mutation, luckily it will be consider, it will be silent mutation, or if it will leads to the danger like the missense mutation. Okay, then at that time amino acids can be changed. So in the silent mutation, what happen? The single nucleotide get changed, like the UCA will adenine will convert into the uracil, then UCU will form, and thus you can see here serine. So the serine they have such kind of codons. So there is a single mutation, okay, but in the nucleotide, but also this synthesis of protein, whatever the codon it will, the codon will code for the amino acid, that is serine will remains for the serines, right? So such kind of mutation is called as a silent mutation. The missense mutations, so it's also the single nucleotide change mutation. But what happened here, because here the serine which is having the codon that is UCA, so a, that A will be firstly convert into the will going to change, okay, and it will become the ACA. So it codes for the threonine in case of serine. So that is called as a missense mutation. So this missense mutation, the point mutation, can be also change a codon so that a different protein is specified. A non synonymous change, that is the single cell anemia, it's a good example of this. So what happen here, the single change in the nucleotide which will be responsible for this severe change in the structure of hemoglobin, right? And thus it will responsible for the change in the structure of red blood cells also. Here you can see the normal red blood cells which are circular, but sickle cells they are moon shape, right? Then frame shift mutation. So this occurs when one or more base pairs are inserted in or deleted from the DNA, respectively causing the insertion or deletion of mutations. Right. So you can see here there is a change that is happened during the frame shift mutation. Okay. So this will going to change the all amino acid sequences. So this is about the DNA damage. Then we will see the DNA damage as well as mutation. Then we will see the DNA repair mechanism which is present in our body, right? Every living organism they have such kind of DNA repair mechanism. Either they are prokaryotic or eukaryotic, unicellular or multicellular. First we will see the base excision repair. Okay, so what happen in the excision repairs, the DNA glycosylase, so this enzyme, right? So it repairs the types of damages like the abnormal bases, uracil, hypoxanthine, xanthine, right? They such kind of abnormal bases they can cure. They alkylate the bases in some other organisms. The pyrimidines dimers, they can be also cured by the DNA glycosylase. DNA polymerase first enzyme is also having the ability to cure, right? So here you can see the base excision repair mechanism. Okay, so the base excision repair mechanism system in the E. coli, right? So this repair is catalyzed by the concerted action of base specific glycosylase. So this enzyme is very important here, it plays important role. Then ap endonuclease DNA polymerase and ligase. So this enzyme work for the curation of such kind of mutation. Okay, so in the base excision repair, an enzyme that is called the DNA glycosylase, it recognize it damaged bases and break the glycosidic linkage between the sugar and the damaged base. So this leaves an apurinic or apyrimidinic sites, that is called as the AP sites, which is a sugar without the base. So this site is then recognized by the AP endonucleases, so which cleave the phosphodiester bonds on the 5' side of the AP site. Okay, the DNA phosphodiesterase. Okay, so this phosphodiesterase or deoxyribose phosphodiesterase then removes the AP sugar phosphate, and then DNA polymerase first it performs the repair synthesis by the degrading DNA in the 5' to 3' direction, whatever you can see here, while filling it with the neo DNA by the template dependent polymerization activity. So finally the DNA ligase seals the nicks. Okay, and the different DNA glycosylations are present in the cell to recognize different kinds of damaged bases. Okay, then we'll see next kind of the DNA repair mechanism, that is called nuclear excision repair. So this nuclear excision repair, it's also called as a NER. Okay, so this is used by the cells to repair the bulky DNA damage. Okay, so it's a non-specific DNA damage. Some kind of chemical adducts or UV photoproducts, they cause the damage to the DNA, and they causes the DNA damage in the bulk form, right? So that's why this repair mechanism, that is nucleotide excision repair mechanism, will be get activated, and it is non-specific. So this is the last try. Okay, so nucleotide excision repair system, they remove most of the bulky adducts from the DNA, right? The enzymes you can see here. Okay, enzymes that is called as the exonuclease complex or URABC complex, which has the three proteins URA, B and URC, which are participating in the repair mechanism or repair process. Okay, we will see that this repair mechanism, as it is not a specific DNA repair mechanism. Okay, so there will be no guarantee of covering the whole DNA damage. Okay, this is like the xeroderma pigmentosa. It's a rare autosomal recessive disease. It affected the patients are having the photosensitivity and the susceptibility to the skin cancer. So it is due to the defect in the nucleotide excision repairs, okay, of the DNA damage. So here you can see in this photograph you can see, okay, nucleotide excision repair mechanism, how it going to work. So it removes the bulky DNA. They identify the 13 mer sequences, okay, and by the enzyme the exonucleases. So this exonuclease enzyme found in the E. coli, okay, so it identifies the 13 mer sequences and removed it, and then DNA polymerase 1 will come and it will synthesize the DNA sequences, correct DNA sequences. Or in the humans, the exonuclease enzymes, so it also act on the 29 mer sequences. So they remove the bulk DNA sequences and new DNA synthesized by the RNA polymerase and ligase will fill the gaps. So in this way the repair mechanism occurs. Okay, by the method of nucleotide excision repair. Then we'll see the mismatch repair system. So this mismatch repair system, it's one of that removes any error which are left in the DNA after DNA replication, right? So error in the replication which are going to leads to the mismatch as a wrong base in the incorporated in the newly synthesized DNA. So thus instead of C means instead of cytosine, if the thymine is incorporated opposite guanine, it leads to the guanine thymine mismatch. So this repair system must discriminate between the parental and the newly synthesized strands and removes the mismatch specifically from the newly synthesized DNA. Okay, so these are the different kinds of enzymes which are listed here. Okay, dimethylase as well as the mutase proteins as well as helicase, single stranded binding protein, DNA polymerase third. So these different kinds of enzymes, they are involved in the correction of mismatch. So this mismatch repair system, it's an excision or resynthesis of system that can be divided into 4 phases: recognition of mismatch by the mutase, recruitment of repair enzymes, excision of the incorporated sequences, and resynthesis by the DNA polymerase using the parental strands as a template strand. So here you can see the action of these enzymes, right, to taken for the correction of such kind of mismatch repairs. So here you can see the enzymes, right? 1 minute. Mismatch. Okay. So here the different kinds of enzymes, they get activated here to correct such kind of mutations. So damage which is induced by the DNA damaging agents, a base is modified. Okay, so it will going to change, and thus the glycosylation, it breaks the glycosidic linkage between a sugar and a base. The apurine, that is AP sites, is registered recognizing by the epinuclease, so that will be going to corrected, and the DNA polymerase synthesize the new DNA and ligase will going to seal the next. So such kind of mutation get corrected by the enzyme mutate, okay, as well as the DNA polymerase first and different other enzymes. Then we'll see the recombination. Okay, so maybe you have the question in your mind, what is meant by recombination. So in case the replicating machinery encounters the damage in the templates, then the replication force the stall, and then either of the two following events can be happen. So DNA polymerase leaves a gap in the damaged strands as it fails to incorporate the correct base position in the DNA. Okay, so this gap it needs to be filled. It is done by the daughter strands strand gap repair or recombination repair. So this recombination repair will be explained, okay, thoroughly. So this mutation alters the single genes or small group of genes in an individual, where the recombination redistributes the content of the genome among the various individuals during the reproduction. So recombination is basically involved in the exchange of the genetic material. So there are mainly two types, homologous and transposons. So recombination is mediated by the breakage and the joining of the DNA strands. First we'll see the homologous recombination. Okay. So generally the in the bacteria the homologous genetic recombination is basically a DNA repair process. Okay. So this is activated when the replicating machinery encounters a damage on the templates. So DNA polymer is stable at the machinery encounters, and the damage on the repair can be incorporated, cannot be incorporated, and any base opposite to the damage. So it leaves a gap in the daughter strands and containing the replications. Okay, which heads of the damage. So the gap left in the daughter molecules has to be filled. So this is done by the recombination between the complementary parent strands, okay, and the gap uh, leaves a gap in the parental strands. Okay, so recombination it can be occurs in the highly frequency during the meiosis, so that the process of uh, chromosomes divided to produce the haploid gametes, sperm cells, or ova in the higher eukaryotes. So each gametes having only one member of chromosome pairs. So here you can see how the recombination takes place during the meiosis process. The segment get exchanges here. You can see. So these chromosomes will become the hybrid chromosome as they exchanges their segments. Here also. Okay. So how this double-stranded DNA damage it can be corrected. Okay. Then we'll see the holiday junction model for homologous recombination. So this is another very important topic for the uh, exam also. So holiday junction model for the recombination, homologous recombination, it can uh, you can repair the DNA damage. So in the bacteria, the homologous genetic recombination is basically a DNA repair process, refers to as the recombination repair. So this is activated when a replacing machinery encounters and damage the template. So DNA polymer is third stalls at the side gap in the daughter strands and continuous replication ahead of the damage, so that the gap between the gap left in the daughter molecules has to be filled. So there is there is a this done by the recombination between the complementary parent strands and the gap daughter strands. So this process is catalyzed by the protein, that is REC-A protein. So as a consequence, so it leaves a gap in the parental strands. Okay. However, this can be easily repaired, and the daughter strands they made on its intact and used can be used as a template for the filling the gaps, right? So in this photograph you can see the homologous recombination, how it takes place. Okay, so this cross section symbol it generally called as a holiday junction. Then we'll see the transposition. So transposition primarily involves in the movement of some specific piece of DNA in the genome. Means the specific segment they can be moved from one region to the another region. Right. So the mobile segments of DNA are called as transposon or transposable elements. So this type of transposition uh, there are two in numbers, the DNA transposition or retrotransposition. DNA transposition, some transposons they are capable of direct transposition of DNA to the DNA. So this may occur either by replacing the transposition or conservating the transposition. So DNA transposition is less common in the than the retrotransposition in case of eukaryotes. However, in case of the prokaryotes, The DNA transposons, they are more important than the RNA transposons. So here you can see the generation of a staggered break of transposes, ligation of transposons to protruding end of the targeted DNA, and thus the generation of the targeting or flanking the direct repeats through the repeats also of repeats of gaps. Okay, then we'll see the retrotransposition. So transposition which involve in the RNA intermediate represents the retrotransposition. It's a copy of a DNA which forms a transposition or transposon, which are also called as a retrotransposon. So then by the enzymes reverse transcriptase, the DNA is copied from the RNA, and the newly form of DNA, which is a copy of a transposons, get integrated into the genome. So this integration, they may occur randomly in the same chromosome or different chromosomes. Okay, so you can see here these transposons located there. So after the transcription, DNA, RNA will form, and after that reverse transcription, DNA will form, right? And thus you can see the change in the location, change in the position of this transposon copy, that is called as a retrotransposon DNA. Okay, so today we will stop here. Thank you.